today I will introduce the scGPT, which is a foundation model for single-cell multi-omics data using the generative AI. And this work was published in 2024, last year, but you see the citation number? Oh, so yeah, everybody loves the AI nowadays. So, okay, let's start with the abstract. Find the gap sentence in here. Yeah, I couldn't find the gap sentence. So, but I found the key sentence. So I think that it is important that they just made the foundation model for the foundation model for the single cell data using the generative AI. So yeah, that's why this work, in my guess, that's why it was published in Nature Method. So, yeah, and this is actually scGPT. So, the structure of the scGPT is quite simple. So you have we have a input embedding structure, and then we have multiple mask attention transformer. So we will study the structure of the self-attention, because this transformer is based on the self-attention algorithm. So, and using that, using this architecture, this model can perform the diverse downstream models, such as clustering or inferring the gene network. So, and before we study about the self-attention, let's start with the attention. So let's assume that we have a single query fly token and a key sentence. So key sentence is composed of the 4 tokens, 4 words. So we can convert this token to the vector composed of integer. So integer vector for each token. And then we will get these vectors. This indicate actually vectors. And after multiplying the weights, then we can obtain the tensor. So these boxes actually are tensors. And you can see the 3 different tensors. So Q tensor and K tensor and V tensor. So Q actually means the query tensor, and K is the key, and V is the value. So, yeah. So actually, we want to find some meaning of the flag in this context. To do that, many people use the attention algorithm. So after we obtain the Q and K and V tensor, we multiply Q and K to obtain the score, this score. So if this score is large, then we can think that this Q and this K tensor is highly correlated or strongly related, right? So if this score is a negative value, we can think that this K and Q are not related, right? So after we obtain this score, we can see the negative values here. But it is really tricky to deal with this negative value as a score, right? So to convert this negative value to the positive value, many people apply the softmax function. And after they apply the softmax function, this large value converted to 60, and this negative value converted to small positive number. And then this score should be normalized, because if we do not deal with this large variance, then some value will explain everything. So to prevent that kind of situation, we divided each value by the total sum of these scores. So now we have this normalized score, and then we also have a key value tensor here, right? So this is nothing but the average weighted sum. So we multiply the score, the weight, to the value tensor, and then we add it to get the final result. So these tensor reflect not only the Q information contained in Q, but also the context of the sequence, right? So so then let's expand this attention to the self-attention. So in this situation, in the self-attention, we have input data which is composed of the 4 tokens. And then if we multiply the 3 different weights, then we will get 3 different tensor sets. So let's apply the attention to this first token. Then we will get this output. And then we can apply this pipeline to the other tokens. Then we will get another tensor set. And we will use this tensor set as the input of the next process. So then we will get this another output. So in this way, they want to this algorithm preserve the structure of each token and the context of the sequence. So then, how can we train this model? So we need to update this weight. I think there are various ways to update this way, but one of the most popular strategies using the masking. So what is the masking strategy? So let's pretend that we do not know these two tokens. Then this model should infer this mask token using the rest tokens, right? Then we can understand this. We can think that this model can, this model learned the structure of the sequence. Right? So, yeah. Then when we calculate the output, let's mask these two calculation by changing the score to the 0. So that means we only use this unmarked input to infer this output tensor. Okay, then we can get these two tensors, first two tensors corresponding to tokens. And when we calculate this marked gene, of course we should use this tensor, right? This tensor. So then, but we don't know about the last token. So we will not use this last token when we infer the third token. So like this, we can infer the other mask token state. Yep. Then we can convert this output to the original input using the fully connected network. After recover all the value, and after comparing the prediction value and true value, we can obtain the prediction score. So we should increase this prediction score, right, by updating the weight. So this is the self-attention based transformer structure, basic structure. And how about the scGPT? So let's think the cell vector. Cell vector have multiple digits, right? Each digit corresponds to each cell, each gene, right? So each gene has their name, and each cell also have their state. For example, some cells could be normal cell, some cells could be the cancer cell, right? So there are several information. So we need to convert that information to the embedding vector. So first, we have gene names. So we have multiple gene names, and we will give index to each gene using the integer. And we also have expression value. So they use the very unique pre-processing method. So they divided the whole range, whole gene expression range into the several intervals, and then they converted the values to the rank. So, for example, if you divided the whole gene expression range into 4 intervals, then you will have a first quartile and second quartile range and third quartile range, maximum quartile range, right? In this range, you could find within the values within this range. So we will convert this value to the order of this input. So this is first input, right? First quartile. So we will change this value to 1. This value to 2, 3, 4. To prevent the effect of the outlier, as observation. And then, yeah, we will get this final preprocessed expression, which is integer, which composes of the integers. So, and then last is the condition tokens. So, as I told you, each cell have their own information, right? Own state. So, if we have normal cell, then we can put 1 to every gene. If we have a cancer cell, then we can give a 0 to the every gene. Yeah. And then we will change these each integer into the vector using the embedding layer. So, and so we have, let's say we have m genes. Let's say we have m genes. Then gene token, length of the gene token should be m, and the length of the expression value should be m, right? And the length of the condition token should be m. So we have same dimension for each quantities, and then we will change this integer into the vector. For example, CD4 name token converted to the 74, right? So using the embedding layer, this 74 converted into the vector with the 512 dimension. So that means we have m genes, and we will give additional 512 dimension. Right? So finally, we will get this m by d. D is the 512 dimension, m by d matrices. After multiplying every embedding tensor. So do you have any questions? No? Okay. Then, yeah, so using this different information and separate the embedding layer, we can get the full embedding for each gene. Right? But they add additional token for each cell in front of all the gene, and they use this token as the cell representative vector. So each gene have a vector, right? After embedding, gene has an embedding, and each gene have 512 lengths vector. That means this token also have a 512 vector, dimensional vector, right? So we can use this vector correspond to CLS as the cell representative vector, which explain the cell state. Okay. After the training. So yeah, we have embedding tensor in here, right? So we will use this tensor as the input. of the tensor of the transformer block. So this is initial input, and we will get this as the input of the transformer block, and then we will get the output. Right? And then we will use this output for the post-transformer block. It's input. Okay. Yeah. And each transformer block is nothing but the self-potential. Right? We have input data HL, and then we can calculate the Q and K and V, query and key and value tensor by multiplying the input data and three-weighted vector tensors. And then what is the QK transpose? QK transpose. QK transpose is inner product of the query tensor and key tensor, and we use that quantity as the score, attention score, right? The weights of the value, each value tensor, right? So this is the score, and then we apply the softmax. So this is just the attention score or attention weights, and then we multiply the V tensor. Right? So then, what is the A mask in here? We have to update, we have to minimize this loss function. So this is the last output. This is the last output of the transformer. And after the fully connected layer, we can get the fully recovered input expression. And then we have to minimize original value, difference in the original value and predicted expression, right? But this expression should include it in the unknown gene. Do you remember that we used the masking strategy previously? So A mask tensor looks like this. So each column corresponds to each key tensor, and each row corresponds to the query tensor. So, and anyway, so we use this strategy previously. So in here, we only use a mask gene to infer the next state of the query. So, in here, as you can see, we mask these three genes. Okay. So, and actually this white boxes is 0, and these blue boxes is the negative input value. So what is meaning of this? So if you put the 0 in here, yeah, this is nothing but the attention score, right? So you just use the multiplication of Q and K and apply the supplements. There's nothing. So this is just score. But if we put negative input, do you remember the shape of the soft score? Softmax function. Minus infinite. Softmax at the minus infinity is equal to 0. This score should be 0. Okay. Then anyway, we have to generate predicted gene expression, right? How to do that? So first, this algorithm starts with only with the cell embedding, which is cell representative vector I already mentioned, right? CLS, remember? And then they just pick, I don't know, the randomly or I think that they have some rule to pick the gene, and then they recover without any gene. And then they recovered this gene and this gene, right? This gene, and this gene. And they estimate another gene which is highly correlated with this restored gene, recovered gene. And they take it and they recover using these these two recovered cell gene embedding and one cell embedding vector. And then they recover these two gene expression. They predict and they apply this process iteratively until the recover full expression. Right? Okay. So then let's assume that we have full predicted gene expression, right? And we know that these genes are marked genes, right? Marked gene. And we will use this marked gene to calculate the loss function. Okay. And they add additional step. So during this process, the cell representative vector was changed. Right? They want to improve the quality of the cell representative vector, cell embedding. So how to do that? They use this embedding as the input of the additional process. And then they apply the same process. And then they use this fully recovered, they use the recovered marked gene set to calculate the second loss function. And then they add it. That's it. Yeah, that's the training step for the scGPT. Yeah, and yeah, we get after the pre-training, we will get the full model, right? Pre-trained model, and this pre-trained model can give us this cell representative vector. And I already mentioned that this cell representative vector serve as some kind of latent vector of each cell, right? So after applying the UMAP to this cell cell embedding, and they get this UMAP embedding, and they added some additional classifier to invert the cell time. And they calculate the accuracy of the predicted cell time by comparing the original annotation. And I think it worked well, right? So, anyway, so, but this is pre-trained model, but we want to use my data, right? We want to use my data, and we want to extract some biological pattern from our data set. To do that, they suggest to use the fine-tuning approach. So this is our data, and we're going to use this structure, pre-trained model structure and parameter, but we have to adjust a little bit, adjust the parameter of the pre-trained model. So to do that, they use the 3 loss term. So first loss term is just simple gene expression prediction. So this is our data set, pre-processed data set, and this is predicted one from this model. And we will use this loss function as one of the loss, this term as one of the loss function terms. So, and then they also want to improve the quality of the HC, cell representative vector, of their cell. Right? To do that, they calculate the query tensor using the embedding layer from the pre-trained model. And then they apply this query tensor with the weighted vector, and the cell representative vector to get predicted gene expression. And they calculate another term for the loss function. And then last term is elastic cell similarity. This is just that. They just want to put closer when this cell representative vector show the high similarity. They want to put them closely. If the similarity is different, these two vectors is dissimilar, then they want to separate them. So this is fine-tuning step, and they apply this fine-tuning step using this query data. And they also invert the cell type, and by comparing compared to the original cell type, they calculate the accuracy of the prediction. And they using this performance, they also compared the performance of the scGPT with the other. Then this is that the scGPT is outperformed than the other packages. Okay, so this is another application of the scGPT. So using the CRISPR technology, we can inhibit the gene expression or activate the gene expression, right? So we call this thing as the gene perturbation. And then after applying then they train the model by pairing the normal cell and perturbed cell. And then they train the model to infer the perturbed cell gene expression from the normal cell gene expression. Then blue violin plot indicated through this distribution of each gene. And the pink one is predicted. So I think these two gene expressions are highly correlated. Right? So they quantified this correlation using Pearson correlation, and they confirmed that the scGPT is better than the conventional algorithm. So, so I why I need to explain this swing pattern. So each gene, actually they have a data set with the multiple perturbation using the perturbations of different genes, many genes, about the 100 genes perturbation data they have. And each point actually at the location of the cell representative vector inferred from the perturbed data. So, for example, in here, gray cluster, this all the representative or cell embedding actually come from the CLF1 perturbation experiment. They confirm that these cluster show the high expression in the, of the KLF1. So, yeah, now we know the perturbated gene name and their expression, right? And we're gonna use this relationship to infer the perturbed gene from the gene expression of the cell vector. So let's put the additional cell vector here. Then we will connect this cell and the other representative cell using the KNN, and using the voting strategy, KNN voting strategy, we will count the edge which has come from each cluster. And then we can, that means if we put the gene expression of the cell, then we can infer the perturbed gene name, right? So, yeah, using this strategy, they infer the perturbed gene name, and then they found that their model actually found the true perturbed gene using this strategy and their model. So this is another advantage of the using scGPT. So I think, yeah. So if you obtain cells from the different experiment, even though each cell belongs to the same cluster, this cell could be far each other due to the artifact, due to the experiments different. So in this example, all the cells come from the different donors, right? So yeah, so we want to merge that. We want to remove that kind of effect, batch effect. We call it as a batch effect. We want to remove it. And actually, scGPT can merge, can remove this batch effect. So if you compare to the conventional algorithm, then it outperforms than this one. Although it removed the batch effect, it preserved cell type structure. So it's good. 2. And yeah, they did a lot of benchmark study. So in here, so we have embedding genes, right? Gene embedding layer during the input embedding. So that embedding layer also trained during the pre-training step. So using that gene embedding, we can estimate the distance between different genes. So if these two different genes close enough, then we will connect each other. And they found that they found this cluster from the gene embedding, and actually using this cluster genes, gene set cluster, they infer the cell state or cell functionality using pathway enrichment analysis. If you put the gene name here, then you will get multiple pathway. So using this, by reading this pathway, we can infer the gene state or gene's functionality. Yeah. So if you use the scGPT, then you will get more pathway compared to the conventional co-expression analysis. Okay. I think this is last experiment. And so if we put this control and perturb data to the scGPT, then we can catch the this attention score, right? This attention score, and this is the attention score. And they use this attention score structure to compare the control and the perturbation experiment. After the normalization, this is just normalization step, and they finally obtain these normalized average metrics, and then they just subtract these two metrics to find the difference. In this difference, gene A was significantly changed, right? Or gene A is, I think, gene A is affect to the gene C significantly. So using this analysis, they found that this, I don't know this genes, but DDIT3 affect to the other multiple genes. And yeah, they also mentioned that the DHL, this gene affects the other gene. And all these research is supported by the previous studies. So yeah, that's it. And if you use scGPT, you can find it in the GitHub. Thank you.